Good morning, students. We have studied the skeletal system, the type of joints, all those part of bones and their movement, liver mechanism, everything in the whichever is required to understand the skeletal system of the human being. But skeletal system is attached with the muscles, and for the to understand the proper movement of the body, we must study about the muscles of the human body. And since in your syllabus it is the sliding filament theory is in your syllabus, so before that we will have a brief description about the muscles, what are the basic structure of muscle, how the sliding fiber, what are the components of muscle, and then we'll come to the sliding filament theory. So let's start with the basic structure of the muscles which contain muscle fibers. What is the structure of muscle fiber? What is the composition or components involved in making a muscle? So let's start with this basic structure. In muscles are having thin elongated cells. Thin elongated cells make muscles, and the muscle fibers. These thin elongated cells are termed as muscle fibers, and these muscle fibers, they contain cytoplasm, and the cytoplasm of the muscle fiber is termed as sarcoplasm. Sarcoplasm. It contains a network of membranes. They are membrane bound structures which is present in each cell, that is the endoplasmic reticulum. But here it is present in the sarcoplasm, so it is sarcoplasmic reticulum. It is a membranous structure, network of membrane which is for transportation or packaging of substances, and that is sarcoplasmic reticulum. Next is mitochondria. Mitochondria of the muscle fiber is termed as sarcosomes. Sarcosomes. Mitochondria of the muscle fiber is sarcosome. Next, bound by each muscle fiber is bound by a membrane, and that membrane is sarcolemma. Each muscle fiber contains numerous myofibrils. I am repeating. Basic structure of muscles. The basic structure of muscle is it contains thin elongated cells with cytoplasm. That specialized cytoplasm is sarcoplasm. Cytoplasm of the muscle fiber is sarcoplasm. It is covered by a membrane, that is sarcolemma. The endoplasmic reticulum is sarcoplasmic reticulum. The mitochondria of the muscle fiber is sarcosomes, and each muscle fiber contains numerous myofibrils. This basic structure we will study in the diagrams shown in the figure. Now this is the structure of LS of muscle fiber, where you can see these areas are the dark bands and the nucleus is there. Nucleus, the area where the bands are not appearing or which is lighter in color is termed as the light band. The cytoplasm, the membranous covering here is the sarcolemma. This is the LS of a muscle fiber. Now various muscle fibers are present or enclosed in a myofibril, and or muscle fibers like here you can see that number of myofibrils. These are the myofibrils. They are enclosed in a sarcoplasm or covered by a membranous covering, and a number of myofibrils are present in a muscle fiber. The dark and light colors show the dark band and the light band here. This is the structure of muscle fiber, a muscle fiber containing a number of myofibrils in it. Next to this is the details about part of myofibril. Details about part of here you can see this complete zone, the white, the lighter section. It is the I band. Next to this is the darker area which is the A band. In the I band there is a dark line. So this dark line is of the actin filament. In this area, in the darker section, H zone is present. This one is the Z zone, Z line or Z zone. Between two Z lines, there is the sarcomere. The two Z lines, the area between two Z lines represents the sarcomere. So this is a part of myofibril, in detail showing a sarcomere. Next is the sliding filament theory, or the filaments. To explain this darker section is the zone which is the H zone, the single line. This darker line in the middle is the H zone. This one is the A band. The darker section is always the A band. Lighter section is the I band. I band having a strong darker line, and these two ends are the Z lines representing a sarcomere. So this is the complete structure. As you can see here, we have started with the LS of muscle fiber, then a muscle fiber containing myofibrils, then part of myofibrils in detail showing sarcomere. Next to this is the detailed structure of actin and myosin filaments in a sarcomere. So this is about the detailed structure of the myofibrils. In the diagrams you have studied about the myofibrils, the structure of sarcoplasm or the myofibrils, the A line, Z line, I bands. So this what is the difference between these bands? Why we are naming this I band or A band? So basic components present in these bands, and we are discussing about the structure of a sarcomere. Sarcomere, let me remind you about sarcomere. Sarcomere is a part of myofibril, or the area between two Z lines. The area between two Z lines in a myofibril is termed as sarcomere. It is having three bands, that is I band, A band, H band, and a line, Z line. So and between these two Z lines, the area between these two Z lines is the sarcomere. Here I band is having only the actin filaments. The filaments present in I band is only actin filaments. A band is having actin as well as myosin filament. Actin and myosin both. In I band only actin filament is present. Here actin and myosin filaments both are present. H band has only myosin filament. Z line is a membrane to which actin filaments are attached on both the sides. I band starts after Z line, so that actin filament of the I band is attached on both the sides of the Z line. So this is I band only actin filament, then the A band actin as well as myosin filament. H band it is having myosin filament only, and the Z line is attached with Z line is a membrane on which actin filaments are attached on both the sides. I band it is adjacent to the A band on both the sides. Just next to the A band is the I band on both the sides. H band A band it is present in the center of the sarcomere. So this is the place where it is present, or the location of these bands in a sarcomere. Sliding filament theory was proposed by H.E. Huxley and A.F. Huxley, and according to their observation, they have proposed this theory according to the observations, to the observation given here, and the changes, they have mentioned some observation, the changes or the sequence of changes which result in these observations, and that's why they have named this theory as sliding filament theory. So let's start with the changes or observations. What happens during a contraction and relaxation of a muscles? The muscular movement is all about contraction and relaxation, and during these contraction and relaxation, the changes that occur is the length of the A band remains constant during the relaxation as well as contraction. The length of A band, there is no change in the length of A band, it remains constant. While the length of I band, it keeps on decreasing, or it decreases during the muscular contraction. During relaxation, there is no change in the I band, but during contraction, this I band, it decreases, which results in the contraction of muscles also. Or next is the H zone. H zone, it becomes shorter and finally it disappears during a complete or fully contracted muscles. In a fully contracted muscle, the H zone disappears and a thicker line comes, and that thicker line is termed as M line. Here M line, H zone disappears in a fully contracted muscle and a thicker M line appears. Not appeared, it is appears. Sarcomere. What happens to the sarcomere? Sarcomere is the area between two Z lines, and the overall length of this decreases due to shortening of the I band. Decrease in the I band result in the shortening of the length of the I band, and that shortening of the length results in the contraction of the sarcomere, or decrease in the length of the sarcomere. Next is the sequences or the sequence of the observation which result in these decreases, these shortening of length, these contraction movements. The decrease or increase in the band structure. After that, the sequences which result in the decreased size of these bands, or increase in the length of, or disappearance of the Z line, or appearance of M line. So for this, the sequences are: first is the actin filament slides over the myosin filament and moves inwards towards the center. Actin filament slides over myosin and moves towards inside. That moving towards inside towards the center result in contraction of this phase, or contraction of this zone. So this one. Then second step is the cross bridges. The cross bridges that I have shown in the diagram. The cross bridges of myosin filament. They are made up of myosin filament. They act like a hook. Hook for what? Hook for pulling the actin filament. Hook of myosin filaments pull the actin filaments. So this pulling, or this result in contraction phase of the muscle fibers. Then next what happens, that there is a breakage of these cross bridges, breakage and then reformation. So that breakage and then the rearrangement of cross bridge takes place. Next step. For this, the breakage of these cross bridges requires energy. Energy is always in the form of ATP, and from where this ATP comes is, this ATP is provided by the enzyme ATPase, which is present in the myosin filament. So the energy comes from the myosin filament by the action of an enzyme ATPase. How much energy is utilized is 1 ATP is needed for one complete cycle of a cross bridge. For one complete cycle of a cross bridge, 1 ATP of energy is required. Next one is actin and myosin filaments. They interact with each other. They make a actomyosin complex. Here, they make a actin and myosin filaments. Their interaction forms actomyosin complex at the point of contact where the actin and myosin filaments comes, and this result in pulling the actin filament, pulling of the actin filament over the myosin filament, which I have mentioned here also, that actin filament slides over the myosin filament. So that sliding movement is provided by the interaction or the formation of actomyosin complex during the contraction. These all steps takes place during the contraction phase, and during the contraction each cross bridge first detaches. It detaches itself from what? It detaches itself from the actin filament and then reattaches at another point to bring a sliding movement, and the name of the theory is sliding filament theory, or this sliding filament theory is just because of the sliding movement of actin and myosin over each other. This actin myosin movement is always when they slide over one another in a sarcomere, towards the movement is towards the inside, towards the center, resulting in a contraction phase. The cross bridges of actin act like a hook for the cross bridges of myosin act like a hook for the actin filament. There is breakage and rearrangement of cross bridges. Energy is provided by the ATPase enzyme present in the myosin filament. 1 ATP for one complete cycle of a cross bridge. Your actin and myosin filament at the point of contact form actomyosin complex, and this results in pulling the actin filament. Pulling of the actin filament over the myosin results in the sliding movement, and the Haizer theory for the contraction is given by the H.E. Huxley and A.F. Huxley as the sliding filament theory. So this is all about the muscular movement. In the next class, we'll study about few disorders, muscular disorders, or the disorders in the skeletal system, or some of the movement also. Thank you students for joining today. Now in the sliding filament theory, we have studied that between these two darker sections are the A band, and between the A band there is I band. The middle section of the A band is termed as H zone. These lines, the extreme ends of the A band, these two lines are termed the Z line. Now what happens to these bands or these lines during the contraction phase is, here it is a maximally contracted muscle fiber, where you can see that these area, the H zone is completely disappeared. The filaments, the actin and myosin filaments, they slide over one another, and there is a sliding movement here. H zone disappears and forms the M line in this case, and this length which is of the I band, the length of the I band decreases. A band remains same, but there is the sliding movement of the muscles or the filaments. In the H zone, sliding movement resulting in a thicker middle line, that is the M line. So this is about the filament theory which is proposed by the Huxley.